Aloha and welcome to our video on DNA replication. The goals today is to tell the significance of the Hershey-Chase experiment. We'll explain what semi-conservative replication is, and we'll describe how enzymes are involved in the process of DNA replication. Okay, so when we talk about the discovery of DNA replication, what we're talking about is the Hershey-Chase experiment. And what Hershey and Chase did is they found these bacteria cells and they infected them with a virus called a bacteriophage. And these viruses would then create a change in the cell or in the process of the bacteria. So we knew that there was some genetic information being transferred. At the time, there were two different groups that thought what it could be. Some thought it was a protein that had coded all the information, and the others thought it was these nucleic acids. So what Hershey and Chase did is they set up two different experiments. They had one up here and the other one is down here. And what they did is they used a radioactive tracer. So in the first experiment, they took this radioactive tracer and they attached it to the protein coat of a virus. So they thought that if the proteins in a virus were what had this information molecules that were being passed on, that by doing so, when this bacteriophage was released on a cell and then transferred the genetic material, if it was a protein, then there would be protein radioactivity. But unfortunately, there was no radioactivity in the cell this way. So what it led was the idea is that it was not a protein. Now they had to check to make sure that it was the nucleic acid, in fact. So they ran the second experiment here. And in the second experiment, they put a radioactive tracer in the nucleic acids. And when they released the phages onto the bacteria again, what they noticed afterwards is that radioactivity was actually inside. And what they found out was that it was this nucleic acid, and in fact it was DNA that was where the information was being stored, and then later on this DNA would then be replicated through the cells, and they could track these tracers through future generations and see how it passed on. Now, one of the terms that you're going to hear in this unit is semiconservative replication. What this means is that, quite simply, when we make DNA, when we have to make our two strands of DNA in interphase during the S phase, right before we have mitosis, what we're going to notice is that this DNA, remember it was a double helix made of two strands, is going to break down the middle, and it's going to use each of these strands as a template. So because those base pairs only bond with a certain one, they have that complementary base pair property, it's going to be serving as a template. Will make sure that when it is recreated or when they do duplicate or replicate the DNA, by using one of the strands as its template, it's going to force the other strand to be lined up properly. And that's what we talk about as semi-conservative replication. So how does DNA replicate? Well, we found that there are these origins of replication, and these are sites on the DNA strand where it will seem to unzip, like we can see here, and form what we call these replication bubbles. And within these replication bubbles is where we see this DNA being copied, and the net result is going to be these two DNA molecules. Now, if you remember, we were talking about that conservative replication. Well, here's an original strand. Notice we have that same original strand now serving as a template for a new strand, and then we end up with our two DNA molecules. One is going to be the original strand, and one will be this new strand that was just formed. Okay, so what actually happens in these replication bubbles? What are we seeing here? Remember, we have our replication fork, which is where it's splitting apart, okay, where it goes from being a double strand here to this open DNA replication bubble here, and where formation of our new strands occurred there. Now, there's an enzyme involved. It's called DNA polymerase, and what DNA polymerase does is it goes along the old strand of DNA, the original, the template, and it will add these complementary base pairs to it, and that builds this second strand, this new strand of DNA. Now, we have two different ways that it works. In the upper strand here, what we notice is the DNA polymerase, it's going this way. Okay, it'll open up here and it's just going to make one long molecule as it travels along this way. This is what they call the leading strand. Down below, the direction of replication is going to be a little bit backwards. And what it does is it actually makes small little segments, and then they put those small little segments together. And this is what they call the lagging strand. So the leading strand is going to go and it's going to make one smooth process, whereas the lagging strand it's by chunks, and they'll kind of chunk it together like that. So let's take a little closer view as we go through this process. And to do this closer view, we'll start off with our origin of replication. And remember, our origin of replication was quite simply just a portion of the DNA that says, hey, start making copies here. So in our case, it's going to be right here. Okay, so here is our final product. We now have 2 new DNA molecules. Remember, each of the molecules is going to have its original strand, which it used as a template, and that's what gives it its semi-conservative replication. We'll also have a new strand here, and the new strand is lined up from the DNA polymerase, so there's very few mistakes going through, which is what we want it to do. And we'll notice that both DNA molecules look that way. They both have an original strand, and then they have a new strand attached to it. So that pretty much sums up our video and what we talked about today with DNA replication. As always, the lessons will go in a little more detail. Good luck with the lessons, and we'll see you in the next video.